Okay, guys. This is the lab test review for the fluid and electrolyte and acid-base balance lab that we did a couple of weeks ago. We're going to discuss what pH is, why the balance of pH in the body is so important to normal functioning human physiology, the definition of acidic and basic, the definition of pH, and we'll talk about a few electrolytes as well. So, first question is to define the term pH. And what you have to remember about this operator P here, little p has a definite um, definition in terms of mathematical operations. If we were to write out the, the, the arithmetic definition for pH, it would be the opposite of the log to the base 10 of the concentration of hydrogen cations, which can also be called protons, in molar quantity. Remember, the definition of molar is moles per liter. And so what this indicates, if you look at this equation, let's put an equal sign here, is that as the concentration of protons rises, the pH will drop, and as the concentration of protons drops, the pH will rise. And you can demonstrate this to yourself by just performing this operation on a calculator. If you've got a handheld calculator with you, most calculators are going to have a digital input. So what you can do is, for instance, right now, type in on your calculator the number 10, okay, and then look for a key on your calculator. Most Texas Instruments calculators have it as a double E. Okay, so look for the double E. Okay, and then after you hit the double E key, type in the number 5, and then look for the plus minus key. That's the change of sign key, and make that a -5. Okay, and now hit the equal sign. Okay, and what you'll get is 10 to the -5th. That's how you enter that. Now, if you were to take the log of that value, okay, just hit the log key, then what you'd get is a -5. And since the definition of P is the opposite of the log to the base 10, the answer would be 5. Okay, so for a solution that is 10 to the -5th in protons, the pH is 5. For a solution that's 10 to the -3rd in protons, the pH is 3, and so forth. Okay, that's those arithmetic operations are called raising to a power and taking a log, and they're what are called inverse operations of each other. You have to go back maybe. A good idea might be to go to Khan Academy and type in pH as a topic and pull up the YouTube video on exactly the definition of pH. This is just sort of a brief description here. Okay, next question. Would a solution with a pH of 7.8 be considered acidic, neutral, or alkaline? And the answer would be alkaline, okay, because it's above 7. 7 is considered neutral, and below 7 is considered acidic. Would a solution be more acidic? Which solution would be more acidic, one with a pH of 2 or a solution with a pH of 4? And the answer is the solution with a pH of 2. Now you might say, well, why is that? Well, let's compare the concentrations of protons in these two solutions. A solution with a pH of 2 would have a proton concentration of 1 times 10 to the -2 molar, and a solution with a pH of 4 would have a hydrogen ion concentration of 1 times 10 to the -4 molar. And y'all know that 10 to the -4 is a smaller number than 10 to the -2. As a result, this is the more acidic solution. Okay, in the above question, how many times greater is the proton concentration in the more acidic solution than the less acidic solution? And the answer is 100 times more. Take-home point here is that for any single-digit change on the pH scale, that's a tenfold change in total quantity. Okay, so something with a pH of 2 is going to have 10 times more protons than something with a pH of 3. Something with a pH of 2 is going to have 100 times more protons than something with a pH of 4, and so on. If the pH of a solution changes from 4 to 7, how many fold decrease in acidity has occurred? And the answer is 1,000 times, because if you subtract 7 from 4, you get 3, okay? And remember, for every single unit change in pH, that's a tenfold change, okay? So that would be 10 times 10 times 10, which is 1,000. Okay, next question. What is the pH indicated by the test paper seen here? Okay, so what you do, this is the part of the test paper that did not go into the solution. Okay, the way that you know that is if you look at Hydrion paper coming straight out of the container, it's going to have a light orange cast to it. You dip it in the solution of choice and it turns this color, okay? And what you try and do is match this color to the swatches that are on the side of the container, and the pH of the solution would be 11. What is the pH indicated by the test paper here? And the answer would be 2. Okay, this is the part that was dipped in the solution. This is the part that remained dry. Okay, this is just showing you the pH scale, and this is sort of a graphical illustration of what I was trying to explain to you earlier. Okay, this is showing you the proton concentration in molar, okay, on this scale, and this is showing you the corresponding pH. Okay, so if this makes it easier for you to kind of wrap your mind around, something that has a proton concentration of 1 molar, okay, that would be 10 to the 0, would have a pH of 0. Something that has a proton concentration of 10 to the minus 1 molar would have a pH of 1 to the minus 2, 2, and so forth on down the scale. Notice that the concentration of hydroxide anions is basically the complement of the concentration of protons in the solution. And the reason for this is that there's a very simple equation that expresses now the quantity pH plus pOH for any given solution. Okay, pH plus pOH is always equal to 14. So let's draw that here. Okay, so pH plus pOH is equal to 14. Okay, and that's based on the ionization constant for water, and I'll show you how to use this equation in just a minute. So pH is an expression of hydrogen ion concentration in molar. The symbol for hydrogen ion concentration is these square brackets around H+. The pH is equal to the opposite of the log of the proton concentration, where the proton concentration is expressed in molar. For example, for a solution where the proton concentration is 10 to the minus 4 molar, the pH is going to be 4. Note that on the scale on the left, the relationship between hydroxide and proton values for any particular solution of pH value. The absolute values for their exponents is always going to add up to 14. So if the concentration of hydroxide anions is 10 to the minus 6 molar, then the concentration of protons is going to be 10 to the minus 8th molar. That's just another way of expressing this equation that I showed you up here. Okay, pH plus pOH is always equal to 14. So let's take an example. If the hydroxide concentration of a particular solution is 10 to the minus 9th moles per liter, or 10 to the 9th molar, what is the proton concentration of that solution in moles per liter? Now, there's a couple of ways you could do this problem, you could just look over here, okay, and you would see that the answer is going to be 10 to the minus fifth, okay, molar. The pH as a result would be 5. The other way to do it would be to use this equation, okay. If we use this equation up here, they're telling us that the hydroxide concentration is 10 to the minus ninth moles per liter. Okay, so what that would mean is that the POH in this equation would equal 9. Okay, and so what we'd have to do is solve for pH. Solve for pH. Okay, and so what plus 9 is 14? And the answer is 5. Okay, and so a pH of 5 is equal to a proton concentration of 1 times 10 to the minus fifth. That's the way that I like to work these equations, because I think this is an easier equation to remember than this statement of concentrations down here in the bullet point. Okay, so the pH of solution would be 5 in this case. Okay. An increase in acidity results in a decrease of pH, and a decrease in acidity results in an increase of pH. And if you go ahead and look at the pH scale, take those different values and plug them in, you'll see that that's the case. Okay. Fill in the missing formula in this reversible reaction. Okay, so what we've got here is carbon dioxide loading into water. We're forming a particular intermediate. We want to find out what that is, and that intermediate is going to dissociate into bicarbonate anion and free protons. The missing component here is carbonic acid, and the formula for carbonic acid is H2CO3. H2CO3. Okay. This is an equilibrium reaction, which means that at equilibrium, the ratios of reactants to products is going to remain constant. So if we try to strain an equilibrium reaction, what it will try to do is reset the concentrations of reactants and products to maintain that ratio. Again, the name for HCO3 minus is bicarbonate anion. Okay, and this is the free proton, and this is what ultimately is going to affect the pH. So as this component builds up, okay, the pH will go down. Okay, go down. So for instance, in this equation, if we were to load this system with carbon dioxide, we would cause the pH to drop because we would produce more of these products. However, if we were to extract carbon dioxide, say from this part of the equilibrium, we would consume protons, form carbonic acid, and the carbonic acid would dissociate into carbon dioxide and water. The CO2 would be blown off as you exhale, and as a result, the pH in this direction would go back up. This is one of the ways that the respiratory system adjusts the body's pH. Okay, below is a solution containing carbonic acid. If you were to blow into this solution with a straw, would you drive the indicated reversible reaction to the right or to the left? And the answer is you would drive it to the right. Okay, so what would happen is that the pH of the solution would drop. Okay, and over here we have a solution containing carbonic acid. If you were to add bicarbonate to this solution, would the pH increase or decrease? And the answer is that the pH would increase because we're loading this part of the reaction, so the reaction will attempt to shift again to maintain these ratios. As a result, more water and carbon dioxide will be produced, and the pH of the solution will then go up. Okay. Explain the unit of concentration expressed on this label. This is 0.01 molar hydrochloric acid. Okay, so what do we mean when we say 0.01 molar? That means 0.01 moles per liter of solution. Okay, another way to express this would be 10 millimolar hydrochloric acid. The prefix milli means 1 times 10 to the minus 3. Basically, two ways to express the exact same concentration. What is the name of the acid represented by this formula? And the answer is hydrochloric acid. This is a prominent component of gastric juice, and this is one of the things that helps us digest the macronutrients that come into the stomach from the esophagus. Okay, what would the pH of this solution be? Okay, so if we go ahead and solve this equation, okay, this is essentially 1 times 10 to the minus 3 molar. Okay, so the way you do that, millimolar, 1, 2, okay, I'm sorry, 1 times 10 to the minus 2 molar, my bad. And so if we express this, okay, so let's go to the pen. This here, okay, is the same thing as saying 1 times 10 to the minus 2 molar. Okay. And so we look here on the scale, if you want to do this the easy way. 1 times 10 to the minus 2 molar over in this pH scale is going to correspond to a pH of 2. Now why do I immediately go here in order to assign the pH? Well, because hydrochloric acid is what we call a strong acid. Okay, and what do strong acids do in a water solution? They completely dissociate, which means that they break apart into their component parts. Okay, so if we draw this equation, HCl in water is going to come apart and is going to form free protons and chloride. Because this dissociates completely in water, the concentration here is going to equal the concentration here and here. So if this solution is 1 times 10 to the minus 2 molar, then the concentration of protons is going to be 1 times 10 to the minus 2 molar, and the resulting pH is going to be 2. For a weak acid, it's different because weak acids do not dissociate completely in an aqueous solution, and so they have to maintain an equilibrium. Okay, the solution has a pH of 2. If you placed 10 milliliters of this solution into a beaker and added enough water to the beaker to bring the solution up to a total volume of 100 mils, how much would you have diluted the sample of the original solution? And what you would have done now is done a 1 to 10 dilution. Okay, the new pH of the solution would be 3, and the reason for that is that you have now rendered this solution 10 times more dilute after you've done this by adding the water. Is the solution in this bottle acidic or alkaline? And the answer is alkaline. This is milk of magnesia. It's used to treat dyspepsia, and it's generally the mode of action is to neutralize stomach acid. If we added distilled water to the solution, if added to distilled water, if we took this and added this to a beaker of distilled water, would the pH of the water increase or decrease? And the answer is it would increase because this is a base. Okay, and what do bases do to pH? They raise it. Acids drop pH. They lower it. Is this solution acidic or alkaline? The answer is this solution is acidic. And if we added this to distilled water, it would cause the pH to decrease. Vinegar is acetic acid. It is a weak acid. Would a solution containing this substance be acidic or alkaline? Okay, this is sodium bicarbonate. Okay, when you add sodium bicarbonate to water, it's going to dissociate into bicarbonate anion and sodium. It's the bicarbonate that's going to cause the pH to rise. Okay, so the solution would become alkaline. If this substance was added to distilled water, would it cause an increase or decrease in pH? And it would cause an increase in pH. Okay, this is protein powder. Protein is a physiological buffer. Okay, so the reason that proteins can buffer solutions is because remember that proteins are essentially made up of amino acids that fall into 3 different groups. They can either be charged, they can be polar, or they can be nonpolar. The polar and the charged amino acids have the ability to either donate or titrate, remove protons from an aqueous solution. So when you have a long protein molecule, what you essentially have is a complex buffer system. So what do buffers do? They resist changes in pH. So if you challenge a buffered solution, it will try to react in a way to either lock up protons if it's an acid challenge, or to neutralize hydroxide anions if it's a base challenge. And as a result, the pH won't shift significantly. So here's our question. You've got 2 beakers with equal volumes of liquid in them. Beaker A has distilled water, and B is a protein solution. So we've dissolved this in some water. If we added equal amounts of milk of magnesia to both beakers, which beaker would show the lower pH? Okay, so what does milk of magnesia do to distilled water? It elevates the pH. Okay, distilled water has no buffering capacity. If we added the milk of magnesia to the protein solution, what would happen is that the pH would not change significantly. So the lower pH is going to be in the solution that contains the protein powder. And we've already discussed how protein can affect the pH of a solution. A good thing to do is probably go back in your book in the chapter on biochemistry and take a look at proteins and how proteins are built. And what you'll see is a discussion about the different amino acids and what their properties are. And that'll explain a little bit better to you how it is that proteins can act as buffers. Okay, what we want to look at next is to examine amino acids and protein a little bit and describe how it is that they can act as chemical buffers. So we'll look at how amino acids can act as acids and bases as separate entities, and we'll also look at how proteins can also buffer solutions. So what we have to remember about a buffer is that a buffer has the ability to resist a pH change, which means that when there is an acid challenge to a buffered solution, it has the ability to bind those acids to a group on one of the buffer molecules and take it out of solution, thus reducing the amount of free protons in solution and reducing the acid challenge to pH, which would normally lower the pH. With a base challenge to a solution, what has to happen is that the buffer system will actually donate protons to the solution and neutralize the active species in the base challenge, which is the hydroxide anion, and as a result form water. So what you're looking at here is the structure of a generic amino acid. Amino acids have an amino group which acts as a base at neutral pH. It tends to have a positive charge. They have a carboxylic acid group which acts as an acid at neutral pH, where it tends to have a negative charge. And then they'll also have any of 20 different side chains. And remember that amino acids are grouped according to the chemical composition of their side chains. The side chains can either be polar which means that they tend to associate with other polar molecules or with water. They could be nonpolar, which means they tend to associate with other nonpolar groups or with fatty acids or steroids. Or they can be charged, in which case they have an electrical charge at neutral pH and tend to associate with residues that have the opposite charge or with polar residues. And that largely dictates the shape that a protein will take depending on the sequence of amino acids within that protein, basically by the nonpolar components seeking out the nonpolar groups, the polar components seeking out the polar groups, and the charged components seeking out components of the opposite charge. We go a long way in dictating the secondary structure of the protein, and as a result of that, the tertiary structure. And if there's more than one polypeptide chain, this can have an influence on the quaternary structure. And it's those different shapes that proteins can adopt that determine their function. Basically, they serve as molecular tools, and they have a lot of different jobs inside living cells. Examples would be catalyzing chemical reactions, supplying structural components, and also helping with membrane transport. But let's look for a minute at the amino acid here and note that there is an acidic group, the carboxyl group, and a basic group, the amino group, on every amino acid. And then depending on the class of the side chain, you have perhaps even a further buffering capacity. But let's for the moment just imagine a side chain here that is nonpolar. Let's say that this particular side chain does not adopt a charge at any pH. And let's just look at what happens at an intermediate pH for a typical amino acid. What you see is that the carboxylic acid group will have a negative charge because it will have donated its proton to the surrounding solution at neutral pH, while the basic portion of the amino acid, the amino group, will have accepted a proton which has a +1 charge, and as a result will serve as a base at neutral pH. Now, what happens when we challenge this particular amino acid with a base is that we're going to introduce a species into the solution that will have the ability to bind with protons. Protons are hydrogen cations. And when this happens, the active component of the base, which is the hydroxide anion, will combine with the proton and will form a molecule of water. The state of the amino acid at this point in a basic solution is going to be that the carboxy end of the amino acid will have a negative charge because it will have donated its proton, and the basic part of the amino acid will have no charge because it's also donated its proton. The result will be, again, to restrict the number of hydroxide anions in solution by forming water, and as a result resist the challenge to pH. If we put this amino acid in an acidic situation, then the reverse occurs. What happens in an acidic situation is we have an acid challenge. Acids are defined as donating protons to the solution, and so in the presence of an amino acid, what will happen is that the carboxyl group will accept a proton and have a neutral charge, while the amino group will remain positively charged and retain its proton. So here we see again an ability to restrict the number of free protons in solution, and as a result resist the pH challenge. And so this is how amino acids, independently even of the side chain, can act as a buffer. They have the ability to either donate or to bind protons in the face of an acid or base challenge, and as a result restrict the amount of pH change that you see in the solution. Now, if we're thinking in terms of an actual protein, what we have to do is make an entire polymer of these amino acids, one after the other after the other. And if we imagine that this particular protein has a considerable number of positively and negatively charged side chains, the positively charged side chains at neutral pH tend to have amino groups on them, while the negatively charged side chains tend to be acidic groups. So examples of negatively charged amino acids would be things like aspartic acid and glutamic acid, whereas the positively charged amino acid side chains would tend to be things such as lysine and arginine, just to name a couple. So if you imagine that we have a single protein that contains a considerable number of these charged side chains, you can see how when we challenge that protein solution either with an acid challenge or a base challenge, we'll restrict the buildup of protons in an acid challenge, or we will neutralize hydroxide species in a base challenge, and as a result, the actual change in pH will be minimal. And we can see this illustrated in the diagram below. This is what happens again to an amino acid in an acid environment. This is what happens to it in a basic environment. And either way, the bottom line is that we restrict either the buildup of protons in the case of an acid challenge, we restrict the buildup of hydroxide anions in the event of a base challenge. Okay, when these two substances are mixed together, a gas was given off. If you remember from this experiment in lab a few weeks ago, we added sodium bicarbonate to the white vinegar solution, and it began fizzing. Okay, the gas that was given off was carbon dioxide. This reaction essentially was an example of adding, in this case, acetic acid to bicarbonate, and what we formed is carbonic acid, which dissociated into water and carbon dioxide. The carbon dioxide was evaporated off as a gas, and what we were left behind with is water, and so this is what we call a neutralization reaction. Okay, what essentially we do here is we render the pH of the solution near 7 as a result. So how did this reaction literally work? Well, if you take the reaction at face value, okay, so let's draw acetic acid real quick here. I won't draw in all the atoms here. I'm going to use the organic shorthand. Okay, acetic acid solution. Okay, this is the acetic acid structure. When you add it to water, okay, acetic acid is going to dissociate into acetate. Okay, this is acetate, and it's going to also have free protons. Okay, so the acetate will have a -1 charge. The proton will dissociate, okay, and this isn't drawn that well, but anyway. And then what happens is that we take this solution and we add bicarbonate. Okay, now what is bicarbonate? Bicarbonate is sodium bicarbonate. So if you add sodium bicarbonate to the solution, let's just write bicarb here. It'll dissociate into sodium, which has no effect on pH, and bicarbonate, which is HCO3-. Now what happens as a result of this introduction? Well, what will happen is that the proton and the bicarbonate anion will form carbonic acid, H2CO3. What will the carbonic acid do? It'll dissociate into carbon dioxide, which bubbles off, and water. Okay, so that disappears. And what we have left behind is a neutral solution, okay, a neutralized solution that's going to contain, some acetate. It's also going to contain some acetic acid, all right, but this will be in equilibrium, okay, this will be in equilibrium, and what you'll have is a neutral pH, all right. You'll also have probably a little bit of bicarbonate left over, but the bottom line is that's how the neutralization reaction occurs. We remove the protons, which are going to be the active species, in terms of lowering the pH. As they disappear, okay, as they get taken up as water molecules, then what we're left with is acetate and bicarbonate and water, and that essentially ends up being a neutral solution. Okay, the pH indicator phenolphthalein was added to the solutions in beaker A and B. Which of these solutions is acidic and which of these is alkaline? Now, phenolphthalein is a color indicator. It tends to be reddish purple in basic solutions, and it tends to go to a light pink at neutral and clear in solutions that are below 7. Okay, so the beaker A is going to be neutral to acidic, and the beaker B, in this case, is going to be the basic beaker. Next question. Phenolphthalein was added to the solution in this beaker. If I exhale into the vertical straw, okay, and as a result of that blow carbon dioxide into the container, what will happen to the container? Well, the pH will begin to drop, and as the pH begins to drop, the phenolphthalein will change color from reddish purple to pink to clear. Okay, and you saw me do that when I blew into the phenolphthalein solution that we made in lab. As I bubbled my breath in through the straw, the solution began to clear, and that's because of the change in pH. Okay, we're asking here for a definition of these terms. Okay, what is an acid? An acid is anything that in an aqueous solution will donate protons to that solution and cause the pH to drop. What is a base? A base is anything that will titrate protons from a solution, as a result reducing the proton concentration and causing the pH to rise. What is the definition of pH? pH is the opposite of the log to the base 10 of the proton concentration in molar units in that particular solution. Next question, what is acidosis? Okay, well, metabolic acidosis is what occurs when the body is producing large amounts of organic acids that are not being carried away by the blood effectively. Okay, now what happens in metabolic acidosis is that these organic acids are proton donors. What they will do is donate protons to the aqueous solution, the interstitial fluid, and the blood plasma, and the result will be an accumulation of protons and as a result a reduction in the pH value. Respiratory acidosis is a little bit different. In respiratory acidosis, what happens is that we do not have a respiratory rate sufficient to clear carbon dioxide as it's generated as a result of normal metabolism. And so what happens as carbon dioxide accumulates in the blood is that it will combine with water and form carbonic acid, which will dissociate into free protons and bicarbonate. The result of the free protons is that the pH of the solution will begin to drop. So the way the body corrects for this is to accelerate the respiratory rate. Metabolic alkalosis is essentially a reduction in the number of protons in solution as a result of metabolic activity. The result being that the pH will begin to rise. Respiratory alkalosis is what happens when the respiratory rate is much more rapid than the rate of carbon dioxide production, and the result there is that as we eliminate carbon dioxide faster than it's being made, it's not allowed to accumulate in tissue fluid or in blood plasma. The result is that the pH will begin to rise as well. What is a buffer? A buffer is anything that has essentially resistance to a pH challenge. A pH challenge can come as a result of a strong acid being introduced into the buffer solution or a strong base being introduced into the buffer solution. In either event, what's going to happen is that the strong acid will be transformed into a weak acid, the strong base will be transformed into a weak base, and the resulting change in pH will be negligible. A reversible reaction is one in which products can move to reactants or reactants can move to products because the energy barrier between those two states is minimal. Every reversible reaction is what we call an equilibrium reaction. The ratio of products to reactants in an equilibrium reaction is determined by something called the equilibrium constant, the KEQ. So if you strain an equilibrium, what will happen is that the reaction will compensate for that change in ratio by producing either more of the product or more of the reactant in order for that ratio to be maintained. Okay, what we have on this next page is essentially a flow chart giving you some different pHs, some different carbon dioxide concentrations, and some different bicarbonate concentrations in blood. And what they want you to do here is, for each of these situations, follow this flow chart and determine whether or not this individual is in metabolic acidosis or alkalosis, and whether or not it's compensated or uncompensated. Okay, so this is how you use the flow chart here. Okay, you basically walk through. It gives you the normal pH ranges of plasma between 7.35 and 7.45, normal carbon dioxide concentrations as a result of blood gas, and normal bicarbonate concentrations in the urine. And what we do is we check these different values, and then we go back and we try and figure out what that person's actual state is. Okay, and so the states for the problems in the previous slide are given here. So just take a peek at these and then go back and look at that flow chart. It's in your lab manual, and you'll see how we were able to derive these values. The other thing I'd like you guys to do is to make sure and watch the video on metabolic acidosis and alkalosis that I've included in this folder, and it tells you the origin of how we used that flow chart on the previous page to try and determine the individual's metabolic state. So just listen to that lecture and read over that chart, and you'll see how these values fall out. Okay, that brings us to the end of this lab test review. Please be sure and check out the video that I referenced earlier, and also review this material in preparation for the lab practical. And if you have any questions, just let me know. Thank you.
Hello, my name is Eric Strong, and I am a hospitalist at the Palo Alto Veterans Hospital and an affiliated assistant professor of medicine at Stanford University. This is the first lecture in this course on understanding ABGs, or the arterial blood gas. In this lecture, I will first be giving a brief overview of the course as a whole. Then, I will provide a summary of what an ABG is, how it's obtained, and what information it contains. The audience of this lecture series is intended to be medical house staff and critical care nurses, as well as motivated students in medicine, nursing, and respiratory therapy, all of whom will, at some point, be involved in the care of critically ill patients. I will be assuming only a basic background in physiology, and I will fill in additional relevant details as necessary along the way. I can imagine that you might initially feel that several hours of instruction on the ABG is a bit much. Perhaps you were looking for a 10-minute crash course, or maybe you didn't realize ABGs were such a complicated topic. However, my goal with this course is to provide you with everything you could ever possibly need to know about ABGs in order to take care of and treat complex, critically ill patients. Please note that these lectures are for educational purposes only and are not intended to direct the care of any specific patient. No video can supersede the opinion of an experienced and licensed healthcare professional. Having said that, I hope you find these videos both interesting and useful. Please feel welcome to submit feedback after viewing them. Here is the lecture outline for this course. First, lectures 1 through 7 will focus on some background acid-base physiology and the basic algorithm for identifying pathophysiologic states of acid-base imbalance. Lectures 8 through 12 will review the differential diagnosis for the 5 major categories of acid-base disorders. Lecture 13 will summarize the common general approach to acid-base analysis, while Lecture 14 will review some common pitfalls and practical issues not addressed by the general approach. Lecture 15 will present an alternative approach to the common algorithm, one based on the Stewart model. Lectures 16 through 19 will shift gears and focus on using the ABG to diagnose problems with oxygenation. Finally, the 20th lecture will cover how to put everything you've already learned to use in analyzing complex clinical scenarios that involve simultaneous abnormalities of both acid-base balance and the gas exchange. By the end of this course, you should be able to accurately interpret any ABG, including being able to establish a likely differential diagnosis given a specific clinical scenario. Throughout these lectures, I will be presenting the most commonly employed approach to analyzing ABGs. At times, it may seem particularly protocolized with an established and invariant algorithm, and it may be tempting to take the simple numbers from the ABG and quickly run through a set of rules to get some definitive answer. However, many of the rules we will be using were initially derived decades ago from small studies involving only healthy human volunteers. Therefore, I would caution against interpreting any ABG too dogmatically. In other words, don't forget about the patient in front of you by focusing too heavily on abstract numbers. Although I will be working through dozens of examples during this course, mastery of ABG interpretation will only happen if you practice it on your own. Watching someone else solve problems will be helpful, but it won't be sufficient. Therefore, I would encourage you to intermittently pause these lectures as I go over individual examples and attempt to first reach the answer yourself. The remainder of this lecture will give a very brief grand overview of the ABG. The learning objective is to be familiar with the values that are obtained from the ABG, along with aspects of physiology that each value can help to better understand. The arterial blood gas is a specific collection of lab tests run on a sample of arterial blood, which is most importantly inclusive of the arterial pH, the partial pressure of oxygen, abbreviated PaO2, and the partial pressure of carbon dioxide, abbreviated PaCO2. Here is a patient having an ABG drawn through a puncture in the radial artery, which is the typical location for this, though it can also be drawn through the femoral artery if absolutely necessary. An ABG can also be drawn through an arterial line, particularly in intubated ICU patients, where serial samples can be drawn and compared without the need for additional punctures. The blood is then injected into a small cartridge, which is usually fed into a handheld ABG analyzer. This is a picture of the iStat. one particular model of ABG analyzer, which is one of the most commonly used such devices in the United States. Depending on the exact model of analyzer and on the type of cartridge used, the ABG can provide an array of various measurements. In addition to the three I just mentioned, it also always includes the bicarbonate level as well as the arterial O2 saturation of hemoglobin. What do we do with these values, you might ask? The pH, PaCO2, and bicarb is used in determining the acid-base status of the patient. That is, whether the pH of arterial blood is normal, too acidemic, or too alkalemic. These values are related via the Henderson-Hasselbalch equation, which will be discussed a bit in Lecture 2. Next, the PaCO2 alone tells us about the status of the patient's ventilation. It does this through the alveolar ventilation equation that states the partial pressure of CO2 in arterial blood is equal to the product of the rate of systemic CO2 production and pressure of inspired air divided by alveolar ventilation. Therefore, if the PaCO2 can be measured, the systemic CO2 production is usually assumed based on the situation, and pressure of inspired air is also known, then alveolar ventilation can be calculated. Alveolar ventilation is in turn determined by the respiratory rate, tidal volume, and dead space. Finally, the PaCO2, PaO2, and the O2 saturation tell us about the patient's oxygenation. The alveolar gas equation allows one to calculate the alveolar oxygen tension, denoted here as P big AO2. The difference between the calculated P big AO2 and the measured P little AO2 is an important physiologic parameter known as the AA gradient, which provides important information regarding the adequacy of a patient's alveolar arterial membrane through which oxygen and carbon dioxide diffuse. One last point about these values from the ABG concerns how they are determined by the machine. So the big three, that is the pH, the PaCO2, and the P little AO2, are all measured directly. The bicarb is calculated using the Henderson-Hasselbalch equation, and the O2 sat is calculated using a complicated nomogram relating PaO2 and temperature. So overall, while the ABG basically contains just 5 values, it provides critical information about a patient's gas exchange and systemic metabolic pathways. It is an essential tool for diagnosing and prognosticating an extremely wide variety of pathologic conditions. The next lecture will discuss normal acid-base regulation in the body, knowledge of which will be necessary for understanding how to use the ABG for diagnosing abnormalities of acid-base balance. Hello, this is Eric Strong from the Palo Alto Veterans Hospital and Stanford University. This is the second lecture on understanding ABGs, and the topic is normal acid-base regulation. Although the topic of acid-base physiology is quite complex, I will be limiting this discussion to just information that will be necessary and clinically relevant at the bedside for the diagnosis and management of common acid-base disorders. The specific learning objectives of this talk are as follows. First, to know the role of acid-base balance in normal physiology. Second, to understand the 3 stages of how the body produces, transports, and ultimately eliminates acid. Next, to be familiar with the concept of compensation. And finally, to be familiar with the interaction between acid-base abnormalities and potassium balance. The primary role of acid-base balance in the body is to maintain a stable pH within a narrow window centered at 7.40. This is necessary as most enzymes in the human body optimally function here, and relatively small perturbations in pH can lead to their denaturing and even irreversible inactivation. There are a few notable exceptions to this. For example, the enzyme pepsin, whose function is to break down ingestive proteins, optimally operates at a pH of about 2, which is perfect for its acidic environment in the stomach. However, the vast majority of enzymes in the human body prefer a pH at or very close to 7.40. The clinical consequences of dysregulation of acid-base balance are many. The most important are poor vascular tone, myocardial pump failure, increased risk of arrhythmias, skeletal muscle weakness, secondary electrolyte abnormalities, delirium or coma, and in its most profound stages, impairment of cellular respiration leading to death. The single most important principle of acid-base balance in the body is that the rate of acid production must equal the rate of acid elimination. This process can be broken down into three separate stages. First, acid is produced as a consequence of normal metabolism and occasionally as a consequence of pathologic processes. Second, this acid has to be transported via the blood. Finally, the acid is eliminated via the lungs and kidneys. Let's take a brief look at each stage one at a time. First is the production of acid. Most of us have a gut response that acid being produced in the body sounds bad. This is largely due to the fact that the word acid contains a lot of negative connotations outside of medicine, such as acid rain or its occasional use as a horrific weapon. However, in the body, the production of acid is actually a natural and necessary consequence of normal metabolism. The physiologic metabolism of different types of macromolecules leads different acids as byproducts. Breakdown of carbohydrates and fats yield carbon dioxide, a volatile acid. This term volatile means that it vaporizes easily, which is a critical characteristic in order for acid-base balance to be possible, as it allows carbon dioxide, which carries the major acid burden of the body, to be exhaled as a gas by the lungs. Breakdown of protein yields sulfuric acid, and breakdown of phospholipids yields phosphoric acid. These acids are created in much smaller amounts than that of carbon dioxide from the metabolism of carbohydrates and fats. However, because both sulfuric and phosphoric acid are non-volatile acids, they can't be eliminated from the body via the lungs and require a significantly more complicated mechanism in the kidneys for their elimination. Although it is of limited clinical relevance, I'll quickly point out that metabolism of certain foods can actually result in generation of alkali. However, the rate of alkali production is much smaller than that of acid, which neutralizes its effect on acid-base balance. In addition to the normal physiologic formation of acids by the body, there are numerous clinical situations in which pathologic production of acid can occur. This is a rough schematic of how the more common of these pathologic acids can be formed. I won't say anything more about these specific processes at this point, as they will be discussed in more detail in future lectures. Once the body has produced acid, either as part of normal physiologic metabolism or as part of an abnormal pathologic process, it needs to be transported in the bloodstream to the lungs and the kidneys, from where it can be eliminated. Acids could simply be transported by having their dissociated hydrogen ions carried freely in the blood. Unfortunately, this would result in sudden and dramatic swings in pH that would not be compatible with life. Therefore, the use of buffers is necessary. In chemistry, a buffer can be defined as such by the presence of two major characteristics. First, it has to consist of either a weak acid and its conjugate base, or a weak base and its conjugate acid. Second, a buffer has the property of resisting changes in pH. In other words, hydrogen ions can be added or removed from a buffer solution with only minimal change in pH. In the human body, by far the most important buffer is actually the system shown here, which consists of three components in equilibrium with one another. On the far right, there are the bicarbonate and hydrogen ions, which can spontaneously combine into H2CO3, more commonly known as carbonic acid. Technically speaking, the bicarbonate ion and carbonic acid pair is the true buffer. Carbonic acid can be split spontaneously into carbon dioxide and water, but this reaction is very slow in the absence of the enzyme carbonic anhydrase. Note that this sequence of reactions is completely reversible. Because carbonic acid is in much smaller quantities as carbon dioxide, for simplicity, this reaction is often summarized as this. While it is the most important, the bicarbonate system isn't the only buffer. Other extracellular buffers include phosphoric acid and albumin, while intracellular buffers include organic phosphates such as 2,3-DPG and ATP, as well as hemoglobin. While these secondary buffers are physiologically important, in my experience, an awareness of them is usually not of great clinical relevance at the bedside. I hate to bring up the Henderson-Hasselbalch equation because I am concerned it may evoke traumatic memories of high school chemistry. However, it is necessary to spend a few minutes on it in order to demonstrate a critical principle in acid-base physiology. To remind you, this equation states that the pH of a solution is equal to the pKa plus the log of the ratio of the concentration of base to the concentration of conjugate acid. The pKa is the negative log of the equilibrium constant, which is an empirically measured constant that describes the kinetics of a specific chemical reaction. If you're concerned that you don't have a particularly strong background in chemistry, or are for any reason about to become lost, don't worry. This will all make sense in a minute. Now, as I just discussed, the major buffer in the body is the bicarbonate and carbonic acid pair. So let's substitute some numbers into our equation to make it specific for human acid-base regulation. The pKa is an empiric constant, which for this reaction is known to be 6.1. In the event that there are any physical chemists listening, you may remember Henry's law, which relates the concentration of a gas and liquid to its partial pressure. Thus, the concentration of carbonic acid can be substituted with 0.03 times the partial pressure of carbon dioxide, with which it is in equilibrium with the help of carbonic anhydrase, and which can be easily measured with the ABG. The 0.03 here, like the 6.1, is an empirically measured constant. This is an interesting result. We now have an equation which links three easily measured values, the pH, bicarbonate concentration, and partial pressure of carbon dioxide. Thus, measurement of just two of these in any given situation should be sufficient to calculate the third. Let's see how this works out for a person with normal acid-base status. A normal bicarbonate level is approximately 24 millimoles per liter. Of course, it's actually a range of normal values, but since we can't plug a range into the equation very easily, just trust me that 24 is the single most reliable number to use for a quote normal bicarb. The normal partial pressure of carbon dioxide in arterial blood is 40 millimeters of mercury. 24 divided by the product of 0.03 and 40 is equal to 20. The log of 20 is 1.3. And finally, 6.1 plus 1.3 equals 7.4, which as it turns out is the normal value of arterial pH. I love it when physiology and math work so well together. You might now ask, so what? Well, here's the clinical relevance of the Henderson-Hasselbalch equation. Since this equation defines a relationship between arterial pH, bicarbonate concentration, and the partial pressure of CO2 in arterial blood, it is impossible to change one of these values without altering the others. In other words, if there is some shift in either the bicarbonate or the PCO2, the pH must necessarily shift as well. Our bodies actually make critical use of this linkage to further help blunt changes in pH due to pathologic processes. This is known generally as compensation. From the equation you can see that in order for the pH to remain constant, any change in bicarb or PCO2 needs to be accompanied by a proportional change in the same direction as the other variable. For example, if the initial pathologic process is too much bicarb, the body responds in such a way as to increase PCO2. Low bicarb triggers a decrease in PCO2. Conversely, if the initial problem is a PCO2 that is too high, the body increases bicarb, whereas a low PCO2 triggers a decrease in bicarb. Although it would seem to be physiologically advantageous to do so, mechanisms of compensation never work perfectly. That is, they never bring the pH completely back to normal. From a physician's standpoint, it's actually good that compensation doesn't return pH all the way to normal, because if it did, it would be practically impossible to diagnose acid-base disorders. That will seem a little more intuitive when we discuss compensation in detail in a future lecture. Now that I've talked a little bit about how the body transports acid in the blood, I will finally talk about how acid is eliminated from the body altogether. As already mentioned, acid is eliminated from the body at two sites. First is the lungs. The mechanism for acid elimination here is the expiration of carbon dioxide. Of course, CO2 itself isn't technically an acid. However, because of the rapid equilibrium it's in with the bicarbonate and hydrogen ion pair, as CO2 is eliminated from the body, formation of new CO2 from the ion pair via carbonic anhydrase becomes kinetically favorable. The second and more complicated site of acid elimination is the kidneys, where acid is partly eliminated in both the proximal convoluted tubule of the nephron as well as the distal tubule and collecting duct. In the former, the major mechanism is through reabsorption of bicarbonate. In the latter, two mechanisms are seen. The distal tubule and collecting duct excrete hydrogen ions into the tubule lumen, where it combines with HPO4 to form phosphoric acid. Phosphoric acid acts as a buffer here in order to prevent the concentration of hydrogen ions in the tubule lumen from becoming so great that the chemical gradient exceeds that which the tubular cells can pump against. The other mechanism present in the distal tubule and collecting duct is the direct excretion of ammonium ion through a complicated series of steps which starts with the metabolism of the amino acid glutamine by the cells of the proximal tubule. Let's take a brief look at each organ. Compared to the kidneys, the lungs' role in acid-base regulation is relatively straightforward. With each inspiration, as oxygen diffuses across the alveolar capillary membrane and into the pulmonary capillary bed, carbon dioxide brought to the lungs by the pulmonary artery diffuses out of the pulmonary capillaries and into the alveoli. From there, the lungs will expel the carbon dioxide with the next expiration. In addition to this role in normal physiology, they can also respond to pathologic changes in pH, as mentioned previously when I talked briefly about the concept of compensation. There are two general pathways by which the lungs can respond to changes in pH: the peripheral pathway and the central pathway. Let's take a look at how the lungs respond during a period of acidemia. Either low arterial pH or high PCO2, which can be sensed independently of one another, stimulate chemoreceptors in the carotid body, which lies right at the bifurcation of the carotid arteries. This is the same location that senses low arterial oxygen tension as well. These chemoreceptors then send neural input to the medullary respiratory center, which responds by increasing minute ventilation. Minute ventilation can be increased by a faster respiratory rate, a larger tidal volume, or both. The net result is a lower PCO2 and thus a higher pH. The primary trigger of the central pathway is low pH in the CSF itself, which stimulates chemoreceptors on the ventral surface of the medulla. These chemoreceptors then stimulate the medullary respiratory center with the same end result. Note that in the presence of alkalemia, the opposite process occurs with the result of increased PCO2 and decreased pH. Moving on to the kidneys, first let me orient you to my perfectly rendered nephron. Up here is the glomerulus, into which flows the afferent arteriole and away from which flows the efferent arteriole. Immediately off the glomerulus is the proximal convoluted tubule, leading to the loop of Henle, and finally ending with the distal tubule and collecting duct, which will eventually lead to the ureters and bladder. After fluid is filtered across the glomerular membrane, the first stop is the proximal tubule, at which point sodium and bicarbonate can be reabsorbed more or less together. Stimuli for this process are low serum pH, hypokalemia, and angiotensin II. While the loop of Henle plays a major role in electrolyte balance and determining the ultimate concentration of urine, its role in specifically acid-base balance is relatively minor and of little clinical importance. Next, we have the collecting duct, where under the stimulus of either aldosterone or low pH, sodium is reabsorbed while potassium and hydrogen is excreted into the lumen. At the same location, under control of hypokalemia, potassium is reabsorbed in exchange for excretion of hydrogen. There is one final aspect of the kidney's role in acid-base balance. Phosphate is usually reabsorbed predominantly in the proximal tubule. However, this reabsorption is blocked by high levels of PTH or parathyroid hormone. Somewhat irrespective of PTH's complicated role in calcium phosphate metabolism, its secretion can also be stimulated by low pH. To what possible advantage is this? Well, if you remember, one of the key components that allows the kidneys to excrete acid in significant quantities is the presence of phosphoric acid in the urine, which acts as a buffer to prevent the urine from becoming too acidic and thus preventing it from having too great a concentration of hydrogen ions for the collecting duct to pump against under the stimulus of either aldosterone, acidemia, or hypokalemia. In other words, blocking reabsorption of phosphate in the proximal tubule provides a greater amount of buffer in the distal tubule and collecting duct. I want you to understand that a professor of renal physiology would likely look at this diagram and be upset with what he or she might perceive as extreme oversimplicity. However, this is all you need to know about the kidney's contribution to acid-base balance at the bedside. The final topic I will discuss in this lecture is the relatively unique relationship between pH and potassium balance. You just saw a little bit about this in the last slide, where I mentioned that hypokalemia can serve as a stimulus for the excretion of hydrogen ions into the urine in exchange for reabsorption of potassium ions. Another interaction between these two ions is related to their transcellular shifts in the presence of acid-base disorders. In the presence of acid-base disturbances, hydrogen ions move across the cell membrane in exchange for potassium. This effect is most pronounced with metabolic acidoses. Once intracellular, hydrogen ions are buffered by organic phosphates and hemoglobin. The consequence is that there may be an apparent abnormality of potassium balance that may not represent the body's total potassium stores. That is, in the presence of acidemia, a patient can be net potassium depleted, but still hyperkalemic based on a serum potassium level. Let's take a closer look at how this works. This blue box will represent a typical cell. And here are a bunch of potassium ions scattered inside and out. The concentration of potassium is greater within the cell, but in this diagram there isn't intended to be any accurate scale of this displayed per se. And then here are some hydrogen ions. So what happens during acidemia? Hydrogen ions start accumulating in the extracellular space. There is then an inward movement of hydrogen ions, and in exchange, there is an outward movement of potassium ions in order to conserve charge. Some of the intracellular hydrogen ions get buffered and no longer directly contribute to the hydrogen ion concentration. Thus, the concentration gradient favors more inward movement of hydrogen and outward movement of potassium. While the newly intracellular hydrogen ions can be buffered, there is no analogous buffering system for newly extracellular potassium ions. Thus, the net result is hyperkalemia. In reality, this process doesn't occur in discrete steps like I just outlined, but rather as a continuous and simultaneous flow. In summary, acidemia leads to hyperkalemia. As a rough guide, for every 0.1 drop in pH, serum potassium can increase an average of 0.6 milliequivalents per liter. Unfortunately, there is a big range seen from patient to patient, which is why potassium levels need to be monitored very closely when correcting severe acid-base disorders. Alkalemia can conversely lead to hypokalemia by the reverse process. There is not a well-described rule of thumb, however, in how to predict the scale of this effect, but it is usually quantitatively smaller than that seen in acidemia. That concludes this lecture on normal acid-base physiology. In the next lecture, I will discuss how to use the ABG to identify simple and uncomplicated acid-base disorders. This is Eric Strong again, and I will be talking to you today about how to identify simple acid-base disorders. Here are the learning objectives of this lecture. First, to understand the difference between acidemia and an acidosis, and between alkalemia and an alkalosis. Second, to know the 4 basic categories of acid-base disorders. And last, to be able to identify simple acid-base disorders. I'll start with the frequently misused terms acidemia and acidosis and alkalemia and alkalosis. The words acidemia and alkalemia refer to a physiologic state dependent solely upon arterial pH. Acidemia is present if the pH is less than 7.35, while alkalemia is present if the pH is greater than 7.45. Acidosis and alkalosis, on the other hand, refer to the individual processes which tend to drive the pH towards acidemia and alkalemia respectively. A patient can be acidemic or alkalemic, but not both. However, a patient can have one or more acidosis and one or more alkalosis, which can directly offset one another. In some cases, this may even result in an overall normal pH. Now I will discuss the 4 basic categories of acid-base disorders, which you may have already inferred from the last lecture. Disorders can be either respiratory or metabolic. Respiratory disorders are those pathologic processes which disrupt acid-base balance due to their effect on the lungs. Specifically, these disorders alter alveolar ventilation. A respiratory acidosis occurs when the PCO2 is too high, that is, the patient is hypoventilating. A respiratory alkalosis occurs when the PCO2 is too low, that is, the patient is hyperventilating. Metabolic disorders are those pathologic processes which disrupt acid-base balance due to their effects on anything other than the lungs. This almost always refers to the kidneys, GI tract, or cellular respiration in general. A metabolic acidosis is present when the serum bicarbonate concentration is too low. A metabolic alkalosis is present when the bicarb is too high. This chart will need to become second nature to you before you are able to master ABGs. You may remember back to the first lecture on this series on the ABG that the 5 primary values given by the ABG are as follows: the pH, the PaCO2, the PaO2, the bicarbonate concentration, and the O2 saturation. Since we are talking just about the acid-base status for now, we will set aside the PaO2 and the O2 sat and just focus on the remaining 3 values. A quick note about the PaCO2: there is a distinction between P(a)CO2 and P(a)CO2, just like there is a distinction between P(a)O2 and P(a)O2. When the A is lowercase, it is referring to the partial pressure of the relevant gas in arterial blood. When the A is uppercase, it is referring to the partial pressure of the relevant gas in the alveolar space. This distinction will be much more important when we talk about a patient's oxygenation status in future lectures. In common usage, however, if someone simply states PCO2 without specifying little or big A, it is assumed to be little a, that is, the partial pressure of carbon dioxide in arterial blood. I will frequently use this shortcut in the remaining lectures. Another quick word about notation concerns the serum bicarbonate level. You may encounter square brackets around the bicarb level like this, and I will intermittently use them during this course. The brackets denote that we are talking about the concentration of bicarbonate ion. These are frequently neglected in informal discussions, and even when I leave them out, I will still be referring to the bicarb concentration in serum unless otherwise specified. So let's get started with actually analyzing the ABG. Now this may seem obvious, but the first step to analyzing the acid-base status of a patient is always to start with the pH. If the pH is less than 7.35, the patient has acidemia and at least one acidosis. Whereas if the pH is greater than 7.45, the patient has alkalemia and at least one alkalosis. Let's look at a quick example. Here, the pH is 7.25. As this is less than 7.35, this patient has an acidemia. That's very straightforward. Next is step 2, check the PCO2. If the PCO2 is deranged in the same direction as the pH, the acid-base disorder is metabolic in origin. If the PCO2 is deranged in the opposite direction as the pH, the acid-base disorder is respiratory in origin. With this example, the PCO2 is 60. This is higher than the normal PCO2 of 40. Since the pH and the PCO2 are deranged in opposite directions, the disorder present is respiratory. Thus, this patient has a respiratory acidosis. That is, the patient has a pathologic process of the respiratory system that is driving the pH to be lower than normal. Let's take a quick look at 3 more examples. In example 1, we have a patient with a pH of 7.50, a PCO2 of 45, and a bicarb of 34. Step 1, look at the pH. 7.50 is greater than 7.45, thus there is an alkalemia. Step 2, look at the PCO2. Both the pH and the PCO2 are higher than normal. Therefore, the disorder is metabolic. That is, this patient has a metabolic alkalosis. Here is example 2. pH is 7.16, PCO2 is 35, and bicarb is 12. Step 1, the pH is low. Therefore, the patient has an acidemia. Step 2, the PCO2 is also low, the same direction as the pH. Therefore, the disorder is metabolic, and we have a metabolic acidosis. Here's the final example. First, the pH is very low, so there's an acidemia. While the pH is lower than normal, the PCO2 is higher than normal. Therefore, the causative disorder is respiratory. Therefore, the patient has a respiratory acidosis. Identifying simple acid-base disorders really is that simple. In the next lecture, I'll discuss further the phenomenon of compensation and how that allows us to identify the presence of more than one acid-base disorder simultaneously. Hello, this is the 4th lecture of this course on interpreting ABGs, and I'm Eric Strong from the Palo Alto Veterans Hospital and Stanford University. Today I will be discussing the phenomenon of compensation and the diagnosis of mixed acid-base disorders. The learning objectives are as follows. First, to understand the basic mechanisms of physiologic compensation as they relate to interpreting ABGs. Second, to be familiar with the specific compensation formulas and their alternatives. Finally, to be able to use an assessment of compensation to identify the presence of a second acid-base disorder. The topic of compensation was first introduced in Lecture 2, but to remind you, the body attempts to compensate for acid-base disorders in such a way as to mitigate derangements of arterial pH. A primary metabolic disorder will result in respiratory compensation. A primary respiratory disorder will result in metabolic compensation. Compensation does not return the pH all the way to normal, and patients never overcompensate. Let's take a quick look at the very basic mechanisms of compensation for the four general types of acid-base disorders. In a metabolic acidosis, the serum bicarb is abnormally low, and thus so is the pH. The body's response to this, that is, its respiratory compensation, is to increase minute ventilation. Increasing minute ventilation lowers PCO2 and thus tries to increase pH back closer to normal. In a metabolic alkalosis, the bicarb is high and thus so is the pH. The body responds by decreasing minute ventilation, which increases PCO2 and drives pH back down somewhat. In a respiratory acidosis, PCO2 is too high and the pH is too low. The body's metabolic response is to absorb more bicarb in the proximal tubule of the kidney and to increase excretion of hydrogen ions in the distal tubule. This increases serum bicarb and helps to increase pH. Finally, in a respiratory alkalosis, PCO2 is too low and the pH too high. The kidneys then reabsorb less bicarb and excrete less hydrogen ions. Before I move on to talk about how we can use the phenomenon of compensation to help diagnose mixed acid-base disorders, there are two other aspects of compensation that I need to quickly mention. First, the speed with which compensation develops after the onset of an acid-base disturbance is very important, as we'll see shortly. How fast do these processes occur? The respiratory compensation for a primary metabolic disorder is measurable within minutes, but may take up to 24 hours to be maximally stimulated, depending upon the circumstances. The metabolic compensation for a primary respiratory disorder takes a bit longer. I've seen estimates of anywhere from 12 hours to 5 days. I think no one really knows for sure because, as with much of acid-base analysis, this time delay is largely estimated from observations in healthy volunteers and may not strictly apply to critically ill patients. The second additional aspect of compensation I need to mention is that the body's compensatory mechanisms for a metabolic alkalosis is relatively poor compared to that of the three other major classes of acid-base disorders. As I recently mentioned, the body compensates for metabolic alkalosis by decreasing ventilation and thus increasing PCO2. If you remember the alveolar gas equation shown here, you might see that by increasing PCO2, P big A O2 will necessarily decrease. That is, the partial pressure of oxygen in the alveolar space will be low, which will result in a lower arterial partial pressure of oxygen. In other words, hypoventilation will necessarily lead to some degree of hypoxia. In patients with normal gas exchange at baseline, hypoxia is a strong stimulus to breathe more. This hypoxia-induced desire to increase one's ventilation significantly blunts the relative hypoventilation driven by metabolic alkalosis. So now how can we use compensation to identify mixed acid-base disorders? By predicting the normal compensatory response to primary acid-base disorders, mixed disorders can be identified. Specifically, if a given patient is not compensating as predicted, a second or even third acid-base abnormality must be present. For example, imagine you have identified the presence of a primary metabolic acidosis in a patient in whom the serum bicarb is 10 milliequivalents per liter. From an equation I'll show shortly, you would predict that the PCO2 should be approximately equal to 23 millimeters of mercury due to respiratory compensation. However, what if the patient actually has a PCO2 measured at 35 millimeters of mercury? What's the possible explanation? A second primary acid-base disorder must be present. In this particular case, since the PCO2 is higher than predicted, it is consistent with the concurrent presence of a primary respiratory acidosis. In order to predict what the expected compensation should be, generations of previous researchers have empirically derived a set of formulas with a different one for each primary disorder. In fact, to make things even more complicated, there is actually more than one formula in common usage for each disorder. The following chart represents the formulas which, in my experience, appear to be the most commonly used in the United States. For a metabolic acidosis, the compensation formula is as follows. PCO2 should be approximately equal to 1.5 times the bicarb plus 8. This is known as Winter's formula, named after Robert Winters, a pediatrician from Columbia University who helped derive it in the 1960s. For a metabolic alkalosis, PCO2 should be approximately equal to 40 plus 0.7 times the difference between measured bicarb and 24. For respiratory acidosis and alkalosis, it depends upon whether the pathologic processes are acute or chronic. Remember, it takes anywhere from 12 hours to 5 days for a respiratory process to become chronic as it pertains to acid-base balance. For an acute respiratory acidosis, the bicarb should be approximately equal to 24 plus the difference between measured PCO2 and 40 divided by 10. For chronic respiratory acidosis, the bicarb should be 24 plus 4 times the difference between PCO2 and 40 over 10. For an acute respiratory alkalosis, bicarb should be 24 minus 2 times the difference between 40 and the PCO2 over 10. And finally, for a chronic respiratory alkalosis, bicarb should be 24 minus 5 times the difference between 40 and the PCO2 over 10. How much variation is allowed for by the approximately equal sign here? For example, it's very common for people to cite Winter's formula as PCO2 equals 1.5 times bicarb plus 8 plus or minus 2. Owing to substantial variation in compensation between different individuals and between different specific acidoses or alkaloses, the confidence intervals for these equations should likely be much wider. One authoritative source cites a plus or minus of 5 millimeters of mercury for PCO2 when estimating respiratory compensation for metabolic disorders, and plus or minus 3 milliequivalents per liter of bicarb when estimating metabolic compensation for respiratory disorders. This wider range of error appropriately reflects the degree of uncertainty we have when diagnosing complicated acid-base disturbances. As I mentioned before, there are some alternative approaches to calculating compensation. I won't go over all of them, but here is one alternative to calculating compensation for respiratory disorders. For an acute respiratory acidosis, bicarb should increase 1 milliequivalent per liter for each 10 millimeters of mercury the PCO2 is above 40. For a chronic respiratory acidosis, bicarb should increase 4 for each 10 the PCO2 is above 40. For an acute respiratory alkalosis, bicarb should decrease 2 for each 10 the PCO2 is below 40. And finally, for a chronic respiratory alkalosis, bicarb should decrease 5 for each 10 the PCO2 is below 40. The astute viewer may recognize that these shortcuts are actually mathematically equivalent to the equations presented on the last slide, but most people find it easier to remember these relationships when it's presented in this manner. There is actually an extremely easy alternative to calculating compensation for metabolic disturbances. In a metabolic disorder with appropriate compensation, the PCO2 in millimeters of mercury is approximately the same as the first 2 digits of the pH after the decimal point. To see what I mean exactly by that, here is a quick example. In this ABG, the pH is 7.27, the PCO2 is 25, and the bicarb is 11. Now take the first 2 digits of the pH after the decimal, which in this case is 27, and compare it to the PCO2. They are approximately equal to one another. Therefore, there is appropriate physiologic compensation for this metabolic acidosis. I've been teaching acid-base disorders to house staff and students for some time, and I initially hated this particular shortcut because it seemed that a trick so simple couldn't possibly be as accurate as necessary. However, I have since conceded that I was wrong on this. Here is a graph where measured serum bicarb in a metabolic acidosis is on the x-axis, and the predicted PCO2 as a result of compensation is on the y-axis. The blue line here represents the PCO2s that are predicted by the shortcut. For example, in a metabolic acidosis with a bicarb of 16, we expect the PCO2 to be about 31. How does this formula compare to predictions from the empirically and scientifically derived Winters formula? I'd say it's pretty darn close, especially once you are dealing with bicarb levels under 20 mole equivalents per liter. How does this shortcut compare to the formal compensation equation for metabolic alkalosis? Here is the shortcut's predictions of PCO2 over a range of serum bicarbs from 24 to 40. And here is the formal equation. This is definitely an approximation that is close enough to use at the bedside. So now let's go through 3 quick examples to see how an assessment of compensation works in practice. Here is example 1, where the pH is 7.50, the PCO2 is 48, and the bicarb is 34. Steps 1 and 2 of acid-base analysis remain the same, checking the pH, and then the PCO2, and then step 3 will be evaluating compensation. So first, the pH of 7.50 is higher than normal, so therefore this patient has an alkalemia and at least one alkalosis. Next, look at the PCO2 of 48. This is higher than the normal PCO2 of 40. Since the pH and PCO2 are deranged in the same direction, the problem here is metabolic in origin and thus a metabolic alkalosis. Now for step 3. We will take a look at the compensation formula for metabolic alkalosis, which states that the predicted PCO2 should be approximately equal to 40 plus 0.7 times the difference between measured bicarb and 24. Is that true in this case? Yes. Therefore, we'd say that this patient has appropriate respiratory compensation. Using the shortcut method, 50 from the pH is approximately equal to 48 from the PCO2, and we reach the same conclusion. Let's move to example number 2. The pH is 7.12, PCO2 is 32, and bicarb is 10. Step 1, check the pH. pH is low, so there is an acidemia. Step 2, check the PCO2. The PCO2 is also low, so the process is metabolic and thus a metabolic acidosis. Step 3, evaluate compensation. For this, we will use Winters formula. So does the PCO2 approximately equal 1.5 times the bicarb plus 8? 32 is not close enough to 23. Therefore, a second disorder is present. Since PCO2 is higher than predicted, this second process must be a respiratory acidosis. Using a shortcut method, 12 from the pH is not approximately equal to 32 from the PCO2, and again we reach the same conclusion that respiratory compensation is not appropriate. Example number 3. In this case, we have a brief clinical vignette. A 24-year-old man is found unresponsive on the floor, soiled with vomit. He was last seen 4 hours prior and appeared well at that time. His ABG is as follows: pH 7.34, PCO2 65, bicarb 34. Step 1. pH is low, so there is an acidemia. Step 2. The PCO2 is high, so there is a respiratory acidosis. Step 3. Evaluate compensation. From the vignette, it seems highly likely that the pathologic process in question is acute in onset. Therefore, we will use our shortcut for an acute respiratory acidosis. Is the bicarb increased by 1 milliequivalent per liter for each 10 millimeters of mercury the PCO2 is above 40? Since the measured 65 is 25 above the normal 40, and 25 is 2.5 times 10, we would expect the bicarb to be 2.5 milliequivalents per liter above normal. You should generally use 24 as a quote normal bicarb for acid-base problems. Therefore, the expected bicarb from this respiratory acidosis would be approximately 26.5. This is significantly different from the measured bicarb of 34. Thus, a second disorder is present. The bicarb is higher than predicted, so the second disorder is a metabolic alkalosis. You might have expected this conclusion from the fact that despite a moderately elevated PCO2 of 65 on the ABG, the pH of 7.34 is remarkably close to normal, and as I've said before, compensation never returns your pH to normal. So that's a little bit about the phenomenon of compensation in acid-base disorders and how to use compensation to identify the presence of an additional acidosis or alkalosis. In the next lecture, I will talk about the anion gap.